Welcome back everyone. I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about Rapoport Luebering cycle or 2,3 BPG shunt. See, as we all know, in glycolysis process, glucose gets converted into glucose 6-phosphate, then fructose 6-phosphate, fructose 1,6-bisphosphate, from there glyceraldehyde 3-phosphate and DHAP. These two are interconvertible. Then glyceraldehyde 3-phosphate gets converted into 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate and ultimately pyruvate. Now, in this process, the 1,3-BPG or the 1,3-bisphosphoglycerate can be converted to 2,3-bisphosphoglycerate by the enzyme phosphoglycerate mutase, where the phosphate group from the first carbon is removed and another phosphate group to the second carbon is attached. Now these 2,3-BPG can also be converted to 3-phosphoglycerate with the help of 2,3-bisphosphoglycerate phosphatase enzyme. Okay. But one interesting thing is to remember that during conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, ATP is formed. But during the shunt pathway of 2,3-BPG, no ATP is formed. So what is the importance of converting 1,3-BPG to 2,3-BPG? See, these 2,3-BPG becomes very important during hypoxic condition in our RBCs. Okay. What is hypoxic condition? Hypoxic condition is then when the partial pressure of oxygen becomes very low, especially at high altitude this happens. Okay. So, in hypoxic condition, what happens? The RBC, we know that RBC contains hemoglobin and hemoglobin has 2 alpha chain and 2 beta chains. Okay. And the 2,3-BPG, this pink circle is very anionic in nature. Now this anionic 2,3-BPG during the hypoxic condition forms ionic bonds with the positively charged beta globin chains. Okay, and they stabilize the short bridge between the beta globin chains. Okay, and as a result, tight form or T form of hemoglobin is formed. In this T form of hemoglobin, the affinity of hemoglobin towards the oxygen is reduced. Okay, so now the T form of hemoglobin during the presence of 2,3-BPG binds less amount of oxygen, rather they unload more oxygen to the tissues. Okay, so the dissociation of oxygen and release to the tissues becomes very faster when 2,3-BPG is attached to it. Okay, and as a result the oxygen dissociation curve shifts to the right. Okay, so the presence of 2,3-BPG during hypoxic condition helps in quick unloading or delivery of oxygen to the tissues, which is very important during low partial pressure of oxygen, and thus they help in high altitude adaptation in hypoxic condition. Okay, now that is the significance of 2,3-BPG in our physiology. Okay. Now another important thing is to remember that in fetal tissues the HbF is present. The F hemoglobin is present, which is made up of 2 alpha chain and 2 gamma chains. But interesting thing is to notice that these gamma chains have very less affinity towards the 2,3-BPG. As a result, they cannot bind to the 2,3-BPG and they cannot unload oxygen as easily as our adult hemoglobin can. Okay, so as a result they has a very high affinity towards oxygen but low dissociation of oxygen to the tissues or low delivery of oxygen to the tissues. Okay, so this opposite thing happens in fetal tissues. Now I think this 2,3-BPG shunt and its importance in our body during hypoxic condition is clear to you. And if you find this video useful, don't forget to hit the like button, share it with your friends, and don't forget to subscribe to my channel for more such videos. Thanks for watching and happy learning.